Hello there. So today we are talking about translation. Normally translation means converting something from one language into another. In biology, when we talk about translation, we're talking about turning the language of DNA into proteins, so also known as protein synthesis. So this is the central dogma of biology, which just means that's the main principle. So this is kind of the key to everything. It's just understanding this process that everything starts with DNA, which contains a code within its sequence of base pairs. DNA gets transcribed into RNA. RNA makes a copy of the code, and then translation is the way of turning that code in the RNA into a real live protein. So DNA controls RNA, which controls the proteins. So proteins, if you remember way back to when we talked about macromolecules, they are polymers of amino acids. Let me move over so you can see this picture here a little better. So what is an amino acid? Well, it's this structure. So every amino acid has an amino end, and aminos have to do with nitrogen. So that means there's a nitrogen group here on one end. The other end is a carboxyl group, which is a carbon with two oxygens. And this oxygen also has a little hydrogen friend over here. So amino end, carboxyl end, and you don't have to remember that. This is just satisfying your curiosity. In the middle is kind of the important difference between amino acids. So all amino acids look like this on the top, and then they have a different chain of atoms that comes off here. So it's just abbreviated R. R means this could be a number of different things. So like amino acid 1 has one type of group here. Amino acid 2 has the same structure at the top, but it's got a different group of atoms hanging off here. When these amino acids get stuck together, the carboxyl group will form a bond with the amino group of the next amino acid, and that they are suddenly called a polypeptide. Just throwing that out there because you'll probably see or hear that a lot. It's used in the book a lot too. So polypeptide just means a chain of amino acids, just because the bond that holds them together is technically called a peptide bond. It's really just a special form of covalent bond. Anywho, so amino acids get put together end to end. These are the monomers. When they get put into chains, they become a polymer. A polymer of amino acids becomes a protein. There are 20 different amino acids that are in your body that are coded for by the DNA by way of RNA. So they have lots of different chains. As you can see here, this is going to show their names and the different chains on them. You do not need to memorize these by any means. I just wanted you to see them. So they have different properties. Some are nonpolar, which means they don't have any charge on them. Some have a positive charge, some have a negative charge, and some have just like a little bit of a positive or negative charge, so they're called polar. Kind of like a water molecule has a polar end. So the oxygen likes to hang on to those electrons a little bit more, giving it one end that's more negative and one end that's more positive. That's all that means. So because of these different properties, these amino acids will interact with each other in different ways to give the protein different shapes and functions, as shown here. So the specific amino acids in a protein or a polypeptide and the order in which they are joined is going to determine ultimately the properties of the protein. So different amino acids, like I said, will interact differently, and they make different shapes. So here's like a helix shape that this chain of amino acids might make. And maybe there's a helix here, and then maybe a different shape over here, maybe another helix somewhere else. So they kind of fold up into this three-dimensional shape. And then they can also join with other polypeptide chains and fold up kind of together into a bigger protein. So that's different ways that the different amino acids here can end up with completely different structures and proteins that do different things, like different enzymes. So it's all due to shape, which is due to the order of amino acids. So how do we get these proteins? Well, the 4 bases that are in DNA, which they get copied into RNA, form a language, which is called the genetic code. The genetic code is read 3 letters or 3 bases at a time, so that each word is 3 bases long, and that corresponds to a single amino acid. So here is a codon. It has 3 different bases in it. This one's G, C, and an A. So anytime there's a G, C, and A together, that is going to code for a single amino acid. So over here behind my picture, it says 1 codon equals 1 amino acid. So that's important to remember, that the secret code is what tells the ribosomes how to build the proteins. Okay, this process is called translation. Oops, sorry, I'm going to move on this side so I'm not blocking anything. Translation is when a ribosome uses the sequence of codons in mRNA to assemble amino acids into a polypeptide chain. So here's an mRNA strand right here. It made a copy of the DNA and it came out into the cytoplasm, and a ribosome attaches to it, and then the ribosome is going to read the genetic code on the mRNA. It's going to read 3 bases at a time, and each of those 3 bases is going to match up with a tRNA. If you remember those from the last lecture, that stands for transfer RNA. These things are attached to one amino acid. Each tRNA has a different amino acid and a different anticodon here, which just means that it's the opposite of a codon, so that the bases will match up. And so this guy recognizes a certain codon on the mRNA and brings that specific amino acid in. So it's a pretty complicated process. We're going to take a look at a video of it in a second. But basically, this mRNA and the ribosome and the tRNAs all work together to get all of these amino acids into a polypeptide chain, all in the right order to make the right protein. All right. Let's take a look at this video that's going to show translation.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of 3 bases in the nucleus. The DNA code determines the order in which the 3 bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
All right, so I hope that kind of put all the different processes together for you. So let's just break it down a little slower. In translation, that's building a protein using the code from an mRNA. It begins when this mRNA in the cytoplasm is going to attach to a ribosome. So here comes one part of the ribosome. It's actually in 2 pieces, but they come together around the mRNA. And the very first transfer RNA that comes in always starts with the AUG. And this one has an amino acid called methionine. So every protein starts with a methionine, because this is the sequence that allows the ribosome to attach. It has to have this one there for this to happen. So the tRNA that carries methionine recognizes that start codon, AUG. It's the first one to bind, and the ribosome binds around it. Step 2, oops, move over. A second tRNA is going to come in carrying its own amino acid. It's going to read the next codon. So there's all these tRNAs hanging around, and the right one will come and match up with the codon of 3 letters. It'll bring its amino acid, and that amino acid will be added to the chain. So what the ribosome is doing is helping these tRNAs find the right spot, and then once they're in place, it's going to catalyze that reaction that attaches the amino acids together. So remember, an enzyme is a biological catalyst, meaning it helps a reaction happen. So this wouldn't happen without a ribosome. These polypeptides would not make a bond on their own, so the ribosome makes that bond happen between the amino acids, and this chain just sort of pops out the top as it builds. So constantly new tRNAs are coming, adding in, and then it just kind of shifts down, adding one after the other. And so once this guy's amino acid has been added to the chain, it's going to fall off and then go find a new amino acid. Then step 3. Oh, my thing's not going to play. Sorry, I had a little GIF. It didn't work. So the polypeptide chain is going to keep continuing to grow until it comes to a stop codon. And there's 2 or 3 different stop codons. But basically, once you get to that codon, then the ribosome knows it's time to stop, and it's going to let go of the mRNA, and it's going to let go of its protein. And then the protein will go off and fold up and become its special self with its specific function. And then the mRNA will go its merry way. And one mRNA can actually make quite a few proteins before it breaks down. So it's not just a one and done thing. So several ribosomes can come and attach at that start codon and go down and read the code and assemble that protein.